Good morning students, welcome to LEELA'S tutorial. So today we will discuss about the applications of animal cell cultures. Mostly they play an important role in biomedical and the clinical research. Right? So instead of sacrificing or creating some harm to the animals, we can work on the animal cells directly in vitro. Right? So let us see what is the first application of animal cell cultures. That is manufacturing. Manufacturing is nothing but productions in bulk. The large scale productions are done by using your animal cells. What productions you are going to do? The production can be your monoclonal antibodies, vaccines, hormones or any other products, biological products. So here let us see what is manufacturing. Manufacturing is the large scale production of viruses for vaccine production. So for vaccine production we need viruses. Then from the viral genome, by using the viruses, we produce the vaccines. Right? So for example, you have polio vaccine, chickenpox, measles, rabies, hepatitis, etc. Right? In the same way here, manufacturing can also be involved in genetically modified products. That means you are modifying the genome such that they produce certain products, right? So which is not existing naturally. You are modifying the genome and allowing the organism to produce that particular protein of interest, right? Example, monoclonal antibodies, insulin, hormones, etc. Right? So here whatever the product you want, that product which codes for particular the gene which codes for particular protein can be isolated, and then we are going to transfer that gene into the animal cell, and then animal cells can be sent for the bulk production. So bulk production, it is nothing but the large scale production where animal cells are cultured in tons. Right? So let us see here one of the example for the bulk production. It is cell culture based vaccine production. So here in case of vaccine production, first you are going to select the tissue. Then from the tissue, by means of disaggregation process, you are going to separate the cells. Right? So here the collagen which is binding all the cells together as a cement has to be diluted or broken. So that is nothing but you are disaggregating the tissue. That means you are separating the cells. Once you obtain the disaggregated cells or the separated cells, you are going to inoculate these cells in animal tissue culture media. So now in the animal tissue culture media, under favorable conditions, right, the pH, temperature, and they are placed in the CO2 incubator where you are going to provide 4 to 5% CO2. Right? So here after achieving all the optimal conditions, you are going to go for the cell culture process. Once the cells are cultured or the cell lines are produced, now you are going to inoculate the viruses into those animal cell cultured plates. Already you have got the cell cultures here. Right? Now the cell cultured plates are inoculated with the viral particles or the viruses. So once the virus infect these animal cells, what happens? They enter into the reproductive stage. Right? So here till now, once they get the host, they utilize the host metabolic mechanism and they undergo division. Okay, the viral particles will be produced and finally it will be the lytic cycle where all the viral particles will be released out. Right? So like that, now you have the animal cells which become host to the viruses. As a result, the virus will be increased in number. Right? So then it is the harvesting of all the viruses which are produced. Once you harvest all the viruses, you send it for the bulk production in order to increase the number of organisms. Then after bulk production, you have to centrifuge all the cellular components and the debris of some other cells. Right? Now after that you are going to go for the purification. purification process. You are going to purify the required product from the cells. After purification process, now you don't want these purified product to change its form or change its activity. So you need to maintain the purified product in the unchanged manner. So what you are supposed to do? You are supposed to add the stabilizers and the preservatives. And apart from that, you are going to add adjuvants also. Adjuvants which enhance the activity of particular vaccine or the medicine, right? So it will increase the activity or it will increase the immune system, right? So here the adjuvants will enhance or increase the activity of that particular medicine. Then after adding all these to the purified product, it is the formulation process. In the formulation process, all the content present in the vaccine will be formulated and it will be sent for the filling process. So now you know the vaccine consists of so and so compounds and the composition of that particular compound. So after that you are going to fill them in the vials. The machine filling is done without any contamination. Everything must be done under sterilized conditions. Then it is the filling. Automatic filling is done by using the machines under sterilized conditions. Once the vials are filled, they are sent for the inspection. So in this inspection, the experts will analyze the quality of the vaccine and also they check for any contamination or impurity or any changes in the products. So here, if the inspection is done, after the inspection, if the product is perfect, it will be sent for the labeling process. So now the vaccine vials, all vials will be labeled with that particular vaccine and what is the dosage and all. Everything, all the information will be present on the label. Once they are labeled, they are sent for the packing. Each package consists of so and so number of vials and what is the name of the vaccine, and then it is released out for the marketing process. Is that clear? So in this way you can go for the manufacturing of any hormone or proteins or anything by using the animal cells. In previously what they used to do, they used to use the animal for the production of these proteins or insulin or anything. But now instead of using the animals, we are using only the cell lines or the cell cultures. Okay, so if you have any doubts in this topic, please put it in comment and subscribe for the further lectures.